Since the beginning of plastic production in the early 1950s, 83 billion tons of plastics have been produced worldwide. These plastics decompose into waste and then into microplastics. Microplastics are everywhere, in food, in drinks such as mineral water, in textiles, and even in the abrasion of car tires. Polyethylene terephthalate, PET for short, polyamide, PA for short, or polyvinyl chloride, PVC for short, are among the best known of the more than 100 different types of plastic. Microplastic particles differ significantly in size, color, and shape. Microplastic particles are getting smaller and smaller. Although they do not decompose completely, at some point they are no longer visible to the human eye. A plastic bag, for example, takes 450 years to decompose into tiny microplastic particles. In order to reduce the negative effects of microplastics on human, animal, and environmental health, binding norms and standards for the analysis of microplastics are needed. The lack of uniform and reproducible measurements and interlaboratory studies exacerbates the situation. Extensive measures are currently being developed to close these gaps. In addition to the creation of policy handbooks, DIN EN ISO 24187 for the characterization of microplastics has already been developed and published. Initial projects such as Plastic Trace, which deals with the traceability of measurement data of nano and microplastics, have already been launched. The use of specially developed materials for the rapid and fully automated analysis of microplastic particles rounds off the activities. i3 track-pore PA membrane with aluminum coating provides high particle contrast between the smooth filter surface and particles in the dark field. The membrane surface is stable over time, even in liquids, and is more cost-effective than gold-coated membranes. But which microscope works best for microplastic analysis? Various ftir, raman, and LDIR microscopes are used to analyze microplastics in the laboratory. However, in order to be able to work efficiently with the extensive and time-consuming analyses, it is advisable to perform the microplastic analysis in the laboratory using an LDIR. The fully automated workflow ensures accurate microplastic characterization, significant time savings, and increased sample throughput. The rapid analysis is achieved, among other things, by duplicating the slides. The aluminum-coated i3 track-pore PA membranes are used individually and remain stable in the slide during the entire analysis. They are firm and vibration-free during the entire process. The results are presented clearly and simply in a short time by the newly developed LDIR software. Although there are many different microscopes for the detection of microplastic particles, the Agilent 8700 LDIR is best suited for this purpose. Together with the i3 track-pore PA membrane, they deliver the most reliable results. Order a membrane sample pack and learn more about the 8700 LDIR microscope.